各位贵宾、师长、同学，大家午安，大家好，我是蝴蝶兰仪礼大使林佩莹，非常高兴能担任今天的司仪。今天的讲座非常荣幸邀请到美国桑纳州立大学 Arizona State University 生物设计研究环境工程终身校董事教授、斯威特环境生物技术中心主任 Dr. Bruce Edward Rittmann 莅临本校。演讲的主题为 The Microorganisms Always Close the Mass Balance， 相信借由本次珍贵难得的机会，能让在座的各位收获满满。Now let's welcome our guests。在讲座开始之前，先向各位介绍今天的与会贵宾，也请各位给予热情的掌声表示欢迎。首先是主讲人 Dr. Bruce Edward Rittmann。Welcome, Professor Rittmann。张兆行副校长 Senior Vice President 张兆庆，黄介成院长丁黄杰成，清大国际学院董瑞安院长，阳明交大林志高教授，亚利桑那州立大学赖彦荣教授，云林科技大学蔡正国研究员，环工系洪俊雄教授，分生所杨文明教授 Professor Yang Wenming， 分生所叶专明助理教授 Assistant Professor Ye Jianming。也感谢生命科学院的师长们拨冗参加。演讲开始前，有请 Professor Rittmann 上台来为本次的演讲海报签名。Before the speech, we invite Professor Rittmann to come to the stage to sign the speech poster. Please come to the stage, Professor. Thank you. Professor, please wait. Please wait. 接着，为了感谢 Professor Rittmann 千里迢迢来到本校进行演讲，本校特别准备一面象征最高荣誉的惠孙琉璃讲座，要致赠给 Professor Rittmann， 请张副校长上台颁发讲座。Professor Rittmann 对环境工程与生物技术领域贡献良多，借由今日合影，象征学界跨领域进一步交流。谢谢副校长，副校长请回座。接着，生命科学院也准备了精美礼品，有请黄介成院长致赠。谢谢黄院长，院长请回座。接下来是分子生物学研究所也准备了精美礼品，有请杨文明教授致赠。谢谢教授，教授请留步。Now we are going to take a group photo. Please, our teachers move the stage to. 请张副校长及现场的师长们上台与教授一同合影留念，纪念这难得的时刻。Thank you and please return to your seats. 谢谢各位师长，师长们请回座。Next, we invite Senior Vice President Chang to deliver the opening remarks for the lecture. 接下来，借由各位的掌声，我们邀请张副校长为讲座进行引言。让我们以热烈的掌声欢迎张副校长。
Good afternoon, Professor Rittmann and faculty members from College of Life Sciences, Dean Huang, and also Dean Yang, and also Professor Yang, and other faculty members from the other universities in Taiwan. Ladies and gentlemen, welcome to Hui-Sun lecture, special lecture today. Hui-Sun special lecture at National Chongqing University is with the highest honor to invite internationally well-known and influential speakers from different societies and academic units around the world. Today is my greatest pleasure to introduce you our honorable speaker, Professor Bruce Rittmann, who is currently Regents Professor, School of Sustainable Engineering and the Built Environment, Arizona State University in the United States of America. Professor Rittmann was elected as academician of National Academy of Engineering in the United States in 2004, and in 2018, he was awarded the prestigious Stockholm Water Prize, the highest honor in the field of water research internationally. It's worth mentioning that the Stockholm Water Prize was jointly established by the Royal Swedish Academy of Science and the Stockholm International Water Institute in 1991 to recognize the substantial contributions of scholars in the water field internationally. Its status is equivalent to the Nobel Prize in this specific field. In addition to these two international awards, Professor Rittmann has also received numerous important honors, including being elected as a fellow of 5 major international academic institutions, such as the AAAS, American Association for the Advancement of Science, ASCE, the American Society of Civil Engineers, IWA, the International Water Association, AAEES, the American Academy of Environmental Engineers and Scientists, and WEF, the Water Environment Federation. Professor Rittmann has also been awarded the Excellence in Environmental Engineering Award from the American Academy of Environment Engineers and Scientists, the Clark Prize for Excellence in Water Research from National Water Research Institute, the BioCluster Award from the International Society for Microbial Ecology and the International Water Association, the John Hodge Quinion Palladium Medal from the American Association of Engineering Societies, the Simon A. Fries Award, and the Walter L. Huber Silver Engineering Research Prize from the American Society of Civil Engineers. Many, many awards, very long lists. The above highest honor are no doubt demonstrating Professor Rittmann's high international visibility and influence in the research field of water. As far as my personal understanding, the research contributions of Professor Rittmann are application of the principles of microbiology, biochemistry, geochemistry, and microbial ecology to the biological treatment of water and wastewater, as well as the biological remediation of polluted water and soil, and eventually apply these techniques to improve human health. Today, Professor Rittmann is going to give us a special lecture which is entitled "The Microorganisms Always Close the Mass Balance." I do believe that the breaking through research ideas from Professor Rittmann's in this field through today's special lecture, not only that we can receive the breaking through research ideas, but also that the researchers and faculty members at National Chongqing University in Taiwan and other universities in Taiwan will identify important research niches to work together for global sustainability in the future. So now, please let us give a big applause to welcome our honorable speaker, academician, Professor Rittmann. Thank you.
Thank you. Okay. Good afternoon. Oh, thank you. I'm very, very happy to be here at Chongqing University. It's my first time to visit this university. I've had a very excellent visit so far, so thank you very much for all those who planned it and invited me. So today, you can see my title, and I'm going to begin by just noting that I am an environmental engineer. My degrees are in environmental engineering, so I'm a real engineer. And so I am a professor of environmental engineering. My degrees are in environmental engineering, so I'm a real engineer. And I'm the director of the Biodesign Sweetie Center for Environmental Biotechnology, in which we use the latest tools from the biological sciences. Sometimes we call it molecular microbial ecology and microbial ecology to do this. So I work at the intersection of engineering and the biological sciences. And this is a great place to work and live. And so my talk today is really going to be about integrating the tools and the concepts from engineering and from all the powerful tools of the biological sciences. So let's begin. Here is a collage of some of the tools that we use in our field. Some of these are historic, some of them are very contemporary. These are tools from what we sometimes call molecular microbial ecology. And I was very, very lucky in my career, because I was the first environmental engineer who was able to utilize these tools. And I was lucky. One day, I was then a professor at the University of Illinois, and in my office, and someone knocked on my door. And I opened the door, and there was this young fellow there, and he said, "I'm a new professor at the university, and I'm looking for people to collaborate. Can I tell you what I do?" I invited him in, and he told me what he did. His name was David Stahl, and if you're into the molecular and microbial ecology business, you know him. He's very famous. And he, at that time, was one of the first to use the first tool, and that's that one, RNA extraction and hybridization. This was the tool from the mid-1980s. It was the very first tool. It allowed us to identify microorganisms by genus and species based on their ribosomal RNA. And he explained what this does, and I'd never heard this before. It was totally new to me. But I said, if what Dr. Stahl tells me is correct, and this works, we will revolutionize our field. So I said, let's work together. And he and I worked together for about 15 years, and we revolutionized the field. Now, everybody who's a leading researcher in the environmental biotechnology uses these tools, and not just this one. In fact, no one uses this one anymore. It's kind of obsolete, but we have all these other ones here, and some that I didn't fit on this. So these tools are very, very many, very, very powerful, and we keep getting more, like almost seems like every week. And what we like to do, of course, is we like to use these in combinations because they give us different types of information about the microorganisms and who they are, what they do, and how they do it. So these omics techniques, all of these things together are the omics techniques. They're very powerful. And they're fabulous. Look at how enthusiastic those people are about omics. They just love omics on this. And everybody loves omics. I love omics. So they're fabulous. But there's a limitation here. And that's going to be part of the theme of my talk today, is how we're going to overcome the limitations of these fabulous tools. They aren't really particularly quantitative. Now, as engineers, we need things to be quantitative. We're going to design a treatment plan or understand the fate of some contaminant. We need to be quantitative. Can't just say, well, you know, it's kind of, we got this organism, we got a few here and a few there, and that doesn't work. You've got to be quantitative. And these tools are not especially quantitative. And I'm giving you an example of not quantitative. It's one very important to my work. The copy number of 16S are DNA operons, the genes for the ribosomal RNA, vary anywhere from 1 to 15 per cell. Now, it's pretty hard to quantify the cells if you have 1 or 2 or 15. And here's a bar chart of this, and you can see the important one is this one down here. So this is one copy number, so there's only one gene. So if you're looking for the gene, there's only one. That's easy. And here's 2, but then you go out here, down here, and some of them are up to 15. And you know, there are a lot of them here. And so if you're trying to quantify, or using the copies of the gene for the 16S ribosomal RNA, and you're trying to quantify it, what do you use? You say there's 1 per cell, or 2 per cell, or 4, or some other number. It makes it really hard to quantify this. And so this tool is incredibly powerful for telling us who's there, but it's not so good for telling us exactly how many are there. And as engineers, we need to know how many. Okay, so one of the things that I see in the field, broadly speaking, the environmental biotechnology field, is that some people make a mistake. It's a big mistake, and that is relying too much on the outputs of these molecular tools. They are really powerful. They give us great information, but they have limitations. And if you rely only on the outputs of these molecular tools, you can fool yourself. One can fool oneself in thinking you know a lot more than you do. And so we can mislead ourselves and we can mislead others. So we don't want to do that. We don't want to mislead ourselves. So we need to do something more than just get an output from some fancy omics tool. Now, the microorganisms never fool themselves. They always get it right. They know who they are and what they're doing. So the microorganisms don't get fooled by omics results, even though we might get confused by them. So what we want to do is we want to be like the microorganisms and never fool ourselves on this. So let's be like the microorganisms. And the key thing is the microorganisms always close the mass balance. They have to. It's their life. They can't make a mistake. It's not like being a student and getting 75% on an exam. They have to get 100% on every exam, every day, every minute. And they do. So they're closing the mass balances. And what are they closing the mass balances on? These are the really important things to close the mass balance on. Electron equivalence. This is how they generate energy. The flow of electrons from electron donor to an electron acceptor generates the energy that allows the organisms to live and do their thing. Nutrients. They need nutrients. It's the building blocks of their little bodies. And they need to get those nutrients. And then acid base. A lot of organisms in their metabolism produce acids and bases. And they need to keep track of that, and we do too. So today, in the amount of time I'm going to do this, I'm going to talk a lot about electron equivalence. It's going to be my #1 mass balance today is electron equivalence. I'm going to talk a little bit about nutrient equivalence because it turns out it's important to one of the stories I'm going to tell you. And I don't have time to do acid-base today. So it's important, but I don't have time today. So I won't do acid-base today. So to help us get a framework, a good framework for the electron mass balance, I have this diagram that I really like. And so what we have here is the flow of electrons from an electron donor substrate. For those of us in the environmental engineering business, if it were an organic, we would call it the BOD, the biochemical oxygen demand. It might be ammonia nitrogen, for example. These are electron donors. They are oxidized. A lot of the electrons then are transferred to an electron acceptor, things like oxygen or nitrate, and it generates some products. But the key thing is this reaction here, this flow of electrons, fraction F sub E, goes here, generates energy. This is the point of this flow. Generate energy for the organisms. That energy is then invested along with the rest of the electrons to produce the biomass. This is what microorganisms do. Actually, this is what we do. We're macroorganisms, but we do the same thing. But it's still simpler for the microorganisms. So this is what we want to know is what's going on here. So where do the electrons flow? How many go to the acceptor? How many go to make the biomass here? And of course, in making the biomass, you're also picking up nutrients like carbon and nitrogen. Here, I'll talk about carbon and nitrogen today. So this is what's going on in all biological cells. They all do this in one way or another. Now, that's a nice simple concept, but in a real environmental biotechnology, you don't have one cell type. Pure cultures are irrelevant to us. There's a lot of them. So you have microorganism 1 doing their own thing, same pattern. You got microorganism 2, same pattern, but doing a different thing, maybe with different acceptors or donors. You got number 3, got number 4, and you probably have maybe 100 of these in a complex ecosystem. So there are a lot of things going on, but all of them are doing it according to this electron balance, like I just showed you, and we'd like to sort it out. We want to know where the electrons go, which reactants, from what electron donors to what acceptors on this. So this is part of it is doing the mass balances, and part of it can be also gained by understanding the results from omics. Okay, so I'm going to illustrate this concept here of integrating the omics results with the mass balances, the quantitation like I just showed you, and I'm going to give you 2 examples today. And it's the power of combining mass balances and the molecular tools. So 2 examples. Example number 1. This is rather a historic example. It was from a paper we published in 1999, so that's quite some time ago. And we didn't have a lot of omics tools in 1999 or the 1990s, but we had some, and we were using them. So this is a paper that was published in Water Science and Technology in '99. I was the first author for some reason I don't remember, but the person who did the real work was Chrissy Laspidou, who was then a PhD student with me. And of course, Chrissy has gone on to become a very successful professor of environmental engineering in Greece. That's her home country. Okay, so let's look at this here. So it was published in '99. And what we did here is we combined molecular and modeling analyses. We combined molecular and modeling analyses, explicitly combined them. You'll see how we did this. It was very powerful. And it's an interesting story as well. Okay, so it was the late 1990s, and our team, by the way, I was then a professor at Northwestern University. Our team had developed the oligonucleotide probes for all of the known nitrifying bacteria of the day. Today we know there's more, but as of 1990s, we knew certain ones, and we sequenced the ribosomes, and we got oligonucleotide probes for every known nitrifier of the 1990s. And then with these probes, we started working with our research sponsor. This is a giant water environment company, worldwide international company. It was then called Société Lyonnaise des Eaux, it's a French company. It's now called Suez Veolia. And they operated wastewater treatment plants all around the world, but we looked at some treatment plants that they had in France and in the Netherlands. And these were treatment plants that had some operating problems that they'd like us to help them solve. So we received mixed liquor samples, that's the biomass, from these processes, from 9 of their treatment plants in these 2 countries here, and we then applied our oligonucleotide probing to look for nitrifiers as well as general active biomass. And these weren't very sophisticated tools by today's standards, but they were the latest and greatest of the 1990s. Also, we received from the treatment plants their operating data. And you'll notice here in this bullet, I say we received the usual limited data. If you go to a real treatment plant, even one operated by a high-class organization like a Société Lyonnaise des Eaux, they don't really take that much data. So their operating data is kind of sparse. And so this is a limitation. But we got what they had. And we got the data from them, and we did the probing, and we knew with our wonderful, powerful new tools that we were going to get great insights into what's going on to these treatment plants, and we were going to help our sponsor operate their plants better. Everybody was going to be really, really happy. And our sponsor, Société Lyonnaise des Eaux, was expecting this. They were expecting a glorious, glorious result from our fabulous brand new molecular tools. So what happened? What was the reality of our study? We accumulated the probing results, and nothing made any sense. It was chaos. Chaos in the data. So I say here, results look like they could have been generated by a random number generator. There's no sense at all to any of the results we got out when we just looked at the molecular probing results. Nothing. And I can tell you, we were disappointed, but our sponsor was very disappointed. Oh yes, they were expecting a glorious silver bullet answer, and what they got was a jumble. And they were unhappy, and we were unhappy because they were unhappy, so we needed to do a better job. So by the way, this is the operating data that we had. I'll just walk through this very briefly here. So there you got your 9 treatment plants over here. There's their name. We knew the volume of the treatment plant, that's well known. We knew the hydraulic retention time, that's how long the water spends in it, that's well known. We can compute then the BOD loading here from this data, that's easy to do. We were given basic influent concentrations, BOD, the organic material, total Kjeldahl nitrogen, TKN, that is the sum of ammonia and organic nitrogen. And then we can compute the ratio. And then we were given a bit of effluent concentrations, the TKN here, MLVSS, this is the biomass concentration in the reactor, and then a measure that they told us of whether they were getting good nitrification or they were not getting good nitrification. And there are 3 of them here, 1, 2, 3, where they were not getting good nitrification, and the operators were very concerned. Okay, so this is the limit of the data we had. Okay, so, and then we had the probing results, and they were just all over. There was no rhyme or reason, no obvious pattern to them. We looked at the ratio of ammonia oxidizers to VSS. This is the total biomass. Huge range, almost a 10-fold range. No obvious pattern. Total active biomass from the probing to VSS, again, a range of about 5. Again, no obvious pattern. Chaos. So, I knew that the microorganisms always close the mass balance, so we better close the mass balance ourselves to get the insight and put that together with the molecular results, and then we might learn something here. So I insisted that we, our team, develop a simple mathematical model for the mass balance that used only data that's actually and normally available, that's what we had, and systematically solved the mass balances on BOD, the organic stuff, and nitrogen, that's what we're trying to nitrify, taking into account two very important metabolic factors. One, heterotrophic bacteria, the ones that are oxidizing the BOD, use nitrogen, ammonia nitrogen, for synthesis to make cells. Nitrifying bacteria, on the other hand, use the nitrogen as their electron donor for energy. So there's a big difference between using nitrogen for synthesis versus using nitrogen for energy. This one gets oxidized, the first one does not get oxidized, it just gets taken up. So we wanted to take this into account in our model so we could understand. So that's where Chrissy came in. Chrissy developed this model on it, and we perfected it. So she did the mathematical modeling analysis, and some of the other people did the molecular analysis, and we wanted to figure out the structure and function of the communities. We published this again in '99. Okay, so I'm going to walk you through some equations in our mass balance here. And how did we use the information that we had at hand to do a mass balance and get the things we needed? So here is an equation for the computation of the XV. This is the total biomass. Total biomass measured as volatile suspended solids close to dry weight. There's a bunch of things up here. This is the synthesis, the net synthesis of the heterotrophic bacteria consuming BOD. This is the net synthesis of the nitrifying bacteria using the change in the TKN. There's always some inert solids that come in. And then you have this ratio of theta x over theta. Theta is the hydraulic retention time, we know that. And theta x is what we call the solids retention time. It's how reciprocal of how fast the cells grow. Now, from the plant data, we knew some of these values. We knew XV, that was measured. We knew theta, that was measured. We knew delta BOD, that was measured. And TKN0, we knew that was measured. So these things in blue, as you can see, they're up in here, they were measured. There are a bunch of parameters here like yields and decay coefficients, and we know those. I mean, we've been studying these organisms for decades. We know what these values are. So we can use them. And then we had to make a very important calculation here. So you have delta TKN. This is the amount of ammonia level nitrogen that's used by the nitrifiers. And it is equal to what's going in, TKN in, that was measured, minus what's going out, that was measured, minus the TKN that the heterotrophs took up for synthesis. Okay? So this is really important, and that's up here. Okay? So we had to subtract out the nitrogen that was going to the heterotrophs and was not available to the nitrifiers. Okay? So from this, with all those numbers and assumptions, we know everything in this equation except theta x, solids retention time, and then we computed it. So this was the independent variable. Okay. Once we got theta x, we could then compute by, again, some mass balances, the concentration, or this is the ratio of ammonia oxidizing bacteria, which were measured by our probes, to total biomass by this. And you can see we needed theta x, we got it. And we can also take the total, all active biomass, which we could measure by our probes, divided by the total biomass, which is the sum of the heterotrophs and the ammonia oxidizers here. So once we got the theta x, we could do these. All right, so we did that. We used the operating data, we made these calculations, and what did we get? So here is a table of our calculations. Again, here in this column here are all the treatment plants. Over here is the calculated solids retention time, theta x. And you can see it's quite a range. This one like 0.42 days. And this one's, 82 days. And, so some of these are really tiny and others are really large. The treatment plant operators didn't make this calculation. We had to do it ourselves. Here are the molecular probing results. The ratio of the probing predicted concentration of the ammonia oxidizers divided by the probing predicted concentration of the heterotrophs. And then this is the ammonia oxidizers divided into the total biomass and total all active bacteria into the total biomass. Okay. So these ones here are what we got from our probes. And this is what our model predicted from the mass balances. Now I've circled a few things. The model predicted that we should have 0 nitrifiers in 3 cases. 1, 2, 3. Okay. 0 nitrifiers. And these happen to be the 3 in the table I showed you earlier where it said no nitrification. So they corresponded. And if you look over here, these 3, they're the ones with the blue circles that had the really low solids retention times. So we were basically just, or not we, the operators were washing out the nitrifiers. Nitrifiers are slow growers, and if you have a short solids retention time, you're going to wipe them out. It's going to wash them out of the system. They're gone. That's what happened here. And you can see if you look at the probing results for these here, the ones that are 0 have very low ratios of ammonia oxidizers to heterotrophs or ammonia oxidizers to total biomass. They're very low. They aren't 0. There's always some background signal, but they're very low compared to, say, these other ones where we had good nitrification. So we could see a clear trend. So now our probing results for the nitrifiers made sense. They all made sense. And if you then look at this is a ratio of we look over here. This is the ratio of the ammonia oxidizers to the heterotrophic bacteria, and this is the model predicted values, and this is the probing results over here. And you can see there's a pretty good correlation here, pretty good correlation of these for all the ones that had nitrifiers, good correlation here. And the key thing that's really key here is that when you go from left to right, this corresponds to increasing influent TKN to BOD. So if you had nitrifiers, you didn't wash them out, then the amount of nitrifiers you had depended completely on the ratio of how much nitrogen was coming in to how much BOD was coming in. If you had a lot of BOD coming in, then you would be down here. You didn't leave much ammonia nitrogen for the nitrifiers to nitrify, and they didn't have many organisms. And if you had a lot of TKN coming into BOD, then you're up here. You got a lot. So it was complete sense, completely logical now. The ratio of nitrogen to BOD determine the relative amounts of ammonia oxidizers. And then if we looked at the total bacteria, this is the ratio of total bacteria, ammonia oxidizers, total active bacteria to total biomass. You can see there's a very strong function here, like this, and this is the probing results, and this is the experimental results. But they correspond well. But the key thing is, as you go from right to left, this is increasing SRT, and those numbers keep going down, 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 because if you leave the biomass in there a long time, that means a long SRT, you get a lot of decay, biomass decay. And the active biomass just decays away, and you don't have much left. So that explained everything here. So when we did all of this, every result was logical. So when we combined the mass balances with the probing results, we achieved great insights. One, for the systems that failed to get nitrification, it was because they had a very short SRT and they were washing out the nitrifiers. Once you had nitrification here, the amount of nitrifiers you had, the ratio of nitrifiers you had to total biomass was controlled by the ratio of TKN to BOD. If you had a lot of TKN coming in, you could have more nitrifiers. Makes complete sense. And the ratio of active biomass was inversely proportional to the solids retention time. Because of the large solids retention time, you just decay away the active biomass. And all this was borne out by the modeling and by the molecular probing results. So, in short, everything was logical. We explained why some plants were doing well, other ones weren't. We explained why the various amounts of nitrifiers and other things were in there. It's completely logical. And I hope that our sponsors were happy. But it was a great paper that we published. I'm very proud of it. Okay, that was my first example. Now I've got a second example, very different example by a group of people from ASU. When I was at ASU. Bubbleless oxygen delivery enabled aerobic benzene activation followed by denitrification coupled degradation. That's a long title. This was a paper published in Biotechnology and Bioengineering in 2019. By 2019, we had many, many more tools. The omics tools had expanded tremendously, and you'll see that reflected in what we did in this study because there were so many more insights we could gain from the molecular tools. But we still had to do the mass balances. Okay, so little background. Benzene. Here's benzene, benzene ring, C6H6. Benzene biodegradation has been very well studied. We know a lot about it. And benzene in aerobic conditions, it's been really well studied, and the initial reactions are oxygenation reactions. So you can have a dioxygenation reaction here. One O2 comes in, it gives you two OHs, or you can have two monooxygenations that each put in one OH, so you end up in the same spot. And then you get a dioxygenation that breaks the ring. And then from here, you just do normal hydroxylation, dehydrogenation reactions, and normal routine catabolism, and you generate a lot of energy from respiration. And you can see here that we're using oxygen here at the beginning for these oxygenation reactions, and we use oxygen here at the for the routine reactions, the respiratory reactions. Okay, so that gives you a little background context. Now, that's great. It works very well, but full aerobic oxidation of oxygen requires a lot of oxygen. You can see here's the stoichiometry for the overall reaction. 1 mole of benzene requires 7.5 moles of oxygen. That's a lot of oxygen. You know, it's expensive to deliver oxygen to an aerobic reactor. It's expensive in terms of money. It's expensive in terms of energy. So we would prefer not to have to deliver a lot of oxygen. So how about we get rid of this costly oxygen delivery here. Oh, I should add, benzene is a volatile compound. So while you're aerating these things, you're bubbling all this oxygen through, you're also stripping benzene out to the atmosphere. It causes smog, it's harmful to humans. So we don't want to volatilize it either. So we would like to minimize the amount of aeration to minimize volatilization, as well as to keep the costs down. Okay, so we would like to not use oxygen, or not use very much oxygen for those reasons. Now, anoxic benzene oxidation is possible. It's been studied, but it's very slow. It's not a very interesting one because of the first steps are very hard. To do the first steps without oxygen is super slow. So, how about a hybrid process in which we use oxygen only for the first steps, for these oxygenation steps. Those are called activation or ring cleavage steps. And then we use another electron acceptor, in our case nitrate for denitrification, for the mineralization, respiration, and growing the cells. So we use This is the first step here. If we use, say, a monooxygenation pathway, now we only use 3 moles. 3 moles of oxygen, not 7.5, but just 3. And then the rest of it, we produce this, this ring cleavage compound here. And you then use nitrate as the electron acceptor. Okay? So only 3 oxygens, and then you use a bunch of nitrate here. And you denitrify the nitrates, reduce the nitrogen gas. So this is what we want to do. Saves a lot of money and environmental risk. Okay, 60% oxygen delivery savings. Good deal. Okay, so let's go back then to this diagram. I showed you the diagram before, and this is exactly what I showed you before. And now what we're going to do is we're going to keep the first part, this part here, we're going to keep that the same because we need the oxygen here for the mono or dioxygenases. And then, but this part, we're going to change 3-nitrate. So we're going to do. So how are we going to do this? So we did a study on this. This is what we published using the oxygen-based membrane biofilm reactor. My group uses membrane biofilm reactors for all kinds of things, and this is one of our examples of them. A membrane biofilm reactor uses hollow fiber gas transfer membranes here. The ones we used here were made by Mitsubishi Rayon in Japan. And we deliver the gas. In this case, we deliver oxygen to the internal inside of the lumen. It diffuses through the wall, and there's a biofilm that grows on the outside of the membranes. And it then uses the oxygen coming from inside out, and it uses the benzene from outside in, and we get these initiation or activation reactions. And it looks like this. This is what the actual reactor looked like after we'd run the experiments. And you can see here are the hollow fibers here on it. And you can see this nice light brown biofilm on there. The fibers, if they're clean, are completely white. Very white. And you can see there's a clear biofilm on here. So we grew a nice biofilm doing this, what I just told you about. Okay? So that's what it looks like. Now the advantage here of using the membrane biofilm reactor is twofold. One, we're delivering what we call bubble-free oxygen. So the oxygen is delivered by diffusion through the membrane wall, so we don't have any bubbles. We're not bubbling it. We're not off-gassing benzene because it's volatile. And the second thing is we can very precisely control the delivery capacity of oxygen by controlling the oxygen pressure inside the membrane. Okay, so we set up the system and we want to answer 3 research questions. Research question number 1. Can we make it work? I mean, does this hybrid approach actually work? Can we get benzene biodegradation where we're only delivering oxygen for the first steps and let nitrate do the rest? If yes, what bacteria are responsible for this? Who's responsible? And then, how are they doing it? What are these initiation reactions, monooxygenations and dioxygenations? What are they actually using? Makes a difference. So, I'll step through our answering of these 3 questions using a combination of mass balances and omics results. All right, so this is the performance of it, and let me walk through this table here. So this is a continuous flow reactor. So we're delivering benzene-containing water continuously all the time. And this down here, this is the duration of the experiment. You can see we ran almost 120 days continuous operation. This up here at the top, you can see the flow rate. And most of the flow rates were around here at about 0.10 mils per minute. Up here, it shows our oxygen delivery. So up here at the beginning, we delivered 100% oxygen, very high oxygen delivery capacity. Here, we cut it to air, so it was only 21%, so about 5-fold less delivery. And then here, we made a special 2% oxygen in nitrogen things. So really get a low oxygen delivery capacity. This one here is the benzene concentration. The black ones are the influent, and open ones are the effluent. Okay. And so you can see the influent was pretty constant, around 100 milligrams per liter benzene. And you can see we were at the beginning, we got essentially 100% removal. And then as we started to decrease the oxygen supply, it went down, but we're still getting good removal all the way through. Okay, so we got good removal. So the answer to the question, can we make it happen in a hybrid fashion, was yes. It worked. We had good benzene removal throughout the duration, no matter what was the oxygen supply capacity here. And then down here on the last one, we're showing here the comparison of the oxygen supply delivery capacity with the amount of electron equivalents that were oxidized. So here you can see really high oxygen delivery capacity, and we were getting complete removal aerobically. We start to cut back, and you can see here and here that the oxygen flux was less than the removal. This is the removal up here. This one here, the light one, that's the removal. And so obviously the organisms weren't using oxygen to remove all of the benzene. They had to be using nitrate. Okay, so where are things going here? So the benzene comes in, it gets activated by these oxygenation reactions, produces short-chain intermediates, like I showed you. They then get oxidized, and the question is, do they get oxidized using oxygen as the electron acceptor, or do they get oxidized using nitrate as the electron acceptor? And we did mass balances by measuring oxygen and measuring nitrate and concentrations in there to see which one was being removed. That's where the mass balances come in here, by measuring the removals of the electron acceptors, so we know how much electron flow went here and how much electron flow went here and here. So we did that. And there was a little bit of electron flow to the biomass. We also could estimate that. Okay, so here we go. This is the most important plot for this example. So I'm going to spend a little time going through what we're looking at here so you can understand what to see in this wonderful plot. Okay, so these are the steady states for the first time. So this is the shortest time, and this is when they're really high oxygen delivery, and then less, and then really low. These circles are the oxygen delivery capacity. So you can see at the beginning we had a really high oxygen delivery capacity, more oxygen than we needed to completely oxidize benzene. But as you see, as we go in time, now it's getting lower and lower and lower. Okay. Now let's look at the plot here. So let's look at the colors first. So the colors are the use of oxygen. For activation, the darker, and for respiration energy, the lighter. You can see when we give them a lot of oxygen, it's roughly 50-50. Okay? Roughly 50-50, maybe a little bit more to the energy reaction. And then this one up here is the electron flow to making biomass. Okay, so give them a lot of oxygen, they make a lot of biomass, and they distribute the electrons roughly equally between activation and respiration. But now you start to restrict the oxygen, and things really change. And let's just shift to the last one here. The last one's easiest to see. Let's look at the very last one. This is where we really had a low DO, low oxygen delivery. And what you can see here, here's the oxygen delivery, and you look down here, and this is the oxygen for activation and cleavage. So they were using essentially almost all the oxygen for those initial activation oxygenation steps. And they used it all up. There was hardly anything left. And then, this one up here is electron flow to nitrate reduction. You can see they couldn't use oxygen, so they started sending electrons to reducing nitrate for respiration. And it became, in fact, the largest. And there was, some biomass synthesis. It's less because the organisms have a lower yield using nitrate versus using oxygen as their terminal electron acceptor. But you can see here, it's very dramatic. Here we give them lots of oxygen. They use lots of oxygen for everything. And here you restrict it. They can only use it for the first steps. And they're forced then to use nitrate for respiration. That's what they do. And they're happy. And we're happy. So it worked. So we got the hybrid system to work. We got it to work exactly like we said. We only use the oxygen for those initial steps, oxygenations, and we don't use oxygen for anything else. But we use nitrate. So then we looked at the distribution of the microorganisms here. Very colorful plot here. The key thing is that there's a lot of the green one here. Let's look at the genus here, genus level. A lot of this green one, which is a Comanodaceae. It's hard to pronounce that. Too many syllables. But so it was about 60% of the biomass was in this Comanodaceae genus. Now, interestingly enough, we seeded the reactor initially with Pseudomonas, because Pseudomonas is famous for being able to degrade all kinds of organics, including aromatics, and they almost disappeared. They're almost gone by the end of the experiment. There are some other ones in here, but again, this Comanodaceae was totally dominant. Okay, so then the question is, that organism was dominant. What was it using for the initial steps? Was it using a dioxygenase, or was it using 2 monooxygenases? And the answer was it was using 2 monooxygenases up here like this. And we know that because that genus has been characterized, its genome sequenced, and we know what the genes are, and it uses monooxygenases. So we were able to then show not only that they did it and who they were, but how they were doing it here using the molecular tools on it. And we also didn't find any genes for anaerobic activation. We didn't expect to find any, and we didn't. That was good. It was all aerobic at the beginning. Okay, so we combine mass balances. We combine this with some more sophisticated genomics analysis, and we learned, 1, we could very carefully control the oxygen supply rate. We could achieve rapid benzene biodegradation with oxygen used only for the initiation cleavage steps, only for those oxygenation steps. And everything after that, all the respiration steps, we're using nitrate. So if we carefully control oxygen delivery, we can get it to work out where we just use the absolute minimum amount of oxygen, our goal. The Comanodaceae was the dominant family, and we know from their gene, the sequence of their genome, that they use initial monooxygenation reactions, and they also can do denitrification. They have the genes for the nitrate respiration, so they have all that in there. So it worked. It worked very well. Strict oxygen control, strict oxygen management in the oxygen-based MBFR can lead to successful treatment of benzene with the minimum use of oxygen, and also no oxygen-based off-gassing. So it was a big practical success, as well as we learned a lot of things from the study on that. And I think with that, we've come to the end. So thank you very much for listening, and I hope you have some penetrating questions. Thanks.
Thank you, Professor Rittmann, for bringing us a wonderful speech. 感谢 Professor 今天精彩丰富的演讲，让我们收获满满。让我们再一次以最热烈的掌声感谢 Professor Rittmann. Now have a Q&A session. Please welcome Dean Huang to moderate our Q&A session.
Okay, we thank Professor Rittmann, very inspiring speech. So is there, now we open for the question. Is there any questions?
Professor Rittmann, it is very interesting to link molecular analysis tool with mass balance using bioenergetics. I wonder if this method can be used in biofilm system, such as attached growth system. Thank you.
Well, the answer, of course, is it can be used everywhere. My second example was the biofilm system, so it clearly worked there. And, you know, the basics of stoichiometry and mass balance are the same for all systems. It really doesn't make much difference. There's kinetic differences. The rates will depend on whether it's a biofilm or not, but the basic mass balances will be the same.
Oh, hi, Professor Rittmann. Just a quick question. Do you think we can recover some electricity during the benzene oxidation?
Okay, that's an interesting question. I like your I like the question. We would have to use something like you know, microbial electrochemical cell. In principle, could do this. In microbial electrochemical cell, the terminal electron acceptor is the anode of a fuel cell, and so in the benzene example, what we would do is to achieve that is you would apply enough oxygen so that you would get the initial steps, like I showed here. And then you've got, you know, simple metabolites. And then they could be oxidized by the microorganisms in the biofilm who have the ability to transfer the electrons to the anode, and then the electrons go from the anode to the cathode, and you can generate electrical current this way. So that's what a microbial fuel cell does. So in principle, you could apply this to it, but your terminal electron acceptor would not be nitrate. It would be an anode of a microbial electrochemical cell.
Do you think it is a feasible way to generate electricity by this method?
Well, yeah, I mean, it's technically possible. I mean, people have, you know, done microbial fuel cells for, you know, a long time. Whether it's practical is another question. And so people have done, you know, we've done a lot of research on microbial electrochemical cells. Our group does a lot on it, and you know, it's scientifically fascinating, and it can be done, but currently the technology is not ready for practical use beyond, you know, small scale in the laboratory. So there's a lot of R&D that would be needed to make this a real world thing. So that's the current status of the microbial electrochemistry. Yeah.
The questions, especially from students. Is there any student want to ask questions?
Yes. By the way, in our group, when we have seminars, we don't allow the professors to ask questions until the students have finished asking their questions. And usually we run out of time, and the professors never get to answer or ask a question because the students have so many questions. So, students, your turn. Okay, there's one. There's a brave one. Okay, I love it. A brave student.
Okay, so thanks for your great talk, Professor. So I have one question is because your research is trying to track where the mass flow to which part of the material. But my question is, in a real situation of the sewage plant, usually have the tens or hundreds of material, they are doing the nutrification for the sewage water. So my question is, how can we track where the mass flow in a material? Yep. By, for example, the first material they break down these nutrients, will break down this sewage water as their biomass. But this biomass might degrade and use again by the second, third, fourth, or five material. Yeah. So how can we still remodel in this situation in the lab?
Great. That's a really, really good question. And it gets to one of the fundamental principles of microbial ecology as we apply it to things like wastewater treatment. And that principle is called functional redundancy. Okay, functional redundancy. So usually the communities that exist in these kind of facilities are quite diverse. You might have hundreds of different unique strains or species out there, and many of them are doing similar things, not exactly the same. And so we're in most cases we're not interested in exactly which species is doing it. We're interested in the group of organisms that can do the same function. And so what we're doing more and more now in terms of using these powerful tools is we're now looking much more at the presence of critically important genes, more so than a particular species. Now, there are a few exceptions to what I just said, but in general, if you're looking at nitrifiers or you're looking at heterotrophic bacteria and BOD, you're not worried about whether it's strain A or strain B. You're worried about that you have organisms that contain the right genes in there. So as we're focusing our attention more, it's going more and more into the presence of functional genes and less towards identifying particular strains. So that's the thing. Now, there are a few exceptions. There are a few very specialized reactions that require, like one organism that can do it. And that's a different story. But in most cases, like in wastewater treatment, there's a lot of functional redundancy, many organisms able to do essentially the same thing.
Okay, thank you, Professor.
Good.
I just have a small question because I'm wondering about the real functional species for that. I think, have you ever tried the, transcriptome or something? Yeah.
Oh yeah. We were now able to do transcriptomics, so we can look not only at what genes are present, but which genes are actually being expressed, and you're at least getting the messenger RNA out there. So yeah, so that's now part of our portfolio. It's becoming easier to do this all the time, and so we're now doing it because you learn. It's good to know what genes are present, but it's even better to know which genes are present and expressed, and then you really have an idea of the functionality of it. So yes, we're really beginning to do more and more of this as the tools evolve and become easier and less expensive to do. Yeah.
Okay, so if there is no further questions, we will close the today's speech. Please thank Professor Rittmann again. Okay, thank you for your attention and coming. Yeah. Thank you very much.
Thank you, Professor Rittmann, for answering our questions. 感谢 Professor Rittmann 为大家解惑，谢谢黄院长，也谢谢师长及同学们的踊跃提问。让我们再一次以最热烈的掌声，感谢 Professor Rittmann 今天的演讲。Thank you for your participation. It is the end of the speech. May you all have a lovely day. 今天的惠蓀讲座到此圆满结束，感谢各位贵宾、师长及同学的热情参与。